All right, so first and foremost, I wanted to ask if anyone has any questions regarding particularly chapters 6 and 7. And I'm actually just going to put the whiteboard up since I'm blurry. Anyway, particularly chapters 6 and 7. So all the parts of the cell, as well as I believe you guys have memory is the other portion. So I would say a couple of tips for studying for this particular one. Um, First and foremost, focus on, for these chapters 6 and 7, focus on the note outline, right? So whatever is in there, add the details from the lecture videos, right? But anything that is not, um, anything that I don't go into super detail on the lecture videos is not something that's going to be on the quizzes. So focus on what's in the note outline. That's enough. There's a lot in this chapter, in these two chapters. I get it. I would remind you though, and I'm sure I did on the videos as well, I would remind you there are whole textbooks that are as thick as yours. I know you don't actually have a physical textbook, but it's about a 2.5 incher. There are whole textbooks that are just called Molecular Biology of the Cell, and chapters, basically they are chapters 6 and 7 in a whole textbook. So this just keeps getting more and more and more in depth. And some of your authors are people who contribute to molecular biology, cellular biology. So they have, when they're asked to write a portion of this general biology textbook, sometimes they kind of get overexcited and they include way more information than can possibly be um, learned in a week or 2 weeks. So that's the point of the lectures, is that you need to read the book and then watch those videos with the note outline and say, oh, she's not talking very much about that. So that must not be very important for the test. Or, my gosh, she spent like 20 minutes on osmosis. That must be really important. Since there are only 2 chapters and there'll be about the same number of questions or maybe even a little bit more, there is more detail in these chapters. So you will be required to know a little bit more detail than what you had to know for 1 through 5, I guess our first ones were. Okay, so anything that is on your mind, particularly that you want to talk about from those chapters?
Yes, the
Okay.
Golgi.
Okay.
I need review of that. And lysosomes.
All right. So the Golgi, usually I think we're still calling it the Golgi apparatus. Fun fact, but not on the test, right? It's the only structure in the cell that is still named for a person, right? So that's just kind of like a little fun fact. And the Golgi's job is multiple things, right? So the Golgi is going to receive, you could call them immature proteins from the rough ER, right? By transport vesicles. And the Golgi has a sidedness to it, remember. So when they draw it, they draw the Golgi so that it looks like this, right? So we draw it that it's kind of like a set of tubes that are, that get smaller, right? And there's a sidedness to it in that it only receives things from the rough ER on one side. And those vesicles, those transport vesicles, are made of phospholipid bilayers. So they just fuse and dump their contents right into the Golgi. So this is the protein. So it's getting dumped right into the Golgi. And then this protein is going to move along the Golgi. It's going to get put into another transport vesicle in order to get to the next layer of the Golgi, the next tube of the Golgi. And that's going to continue to happen sort of back and forth, back and forth, back and forth. And I know I've only drawn like four pieces of the Golgi, but if you look in your book, you'll see a much nicer picture. What goes on in these tubes? Different things in different tubes. Again, there's 2 whole chapters about Golgi, or 3 or 4 chapters in that giant molecular biology of the cell book. We're compressing it down into like 2 sentences, okay? So the Golgi's job is to receive the proteins, and they are in sort of an immature state from the endoplasmic reticulum. And then we say process the proteins, which means lots of things. It means trim off an excess piece. It means add either sometimes a carbohydrate or a lipid, right? So sometimes we get glycoproteins. Glyco would be a carbohydrate added, or a lipoprotein, have a lipid added to it. So the proteins are being processed in that sense. They might be being made into shorter pieces. They might be being made larger. They are also being folded. So remember, the folding starts in the rough ER, but that tertiary quaternary structure to proteins. So that secondary, tertiary, quaternary structure. So alpha helix, beta pleated sheet, that mostly is completed in the rough endoplasmic reticulum. However, the tertiary structure, chemical bonds, as well as putting subunits together to make a protein that has quaternary structure, which we know, like hemoglobin and that transthyretin protein, if they don't have their quaternary structure, they're not actually functional, right? So folding slash levels of protein structure slash organization. I think your book calls it protein structure. And I'm just going to put in parentheses tertiary quaternary, right, structure. So putting the whole protein together. And also, because as if that wasn't enough, then in these levels here, these layers of the Golgi that are the smaller ones, we're also going to be doing the sorting, which is what people always just tell you in, like, 4th grade on. The Golgi is like the post office. The Golgi is like FedEx. They store all the packages and send them out to the different areas of the cell as needed. That's true, but that happens at the end, right? And these functions, this processing and the folding, is actually way more, in some respects, equally as important as the sorting. So by sorting, we mean sorting into vesicles that are destined for the same location inside the cell. So some of these vesicles are going to be going to the mitochondria, because the mitochondria can make its own proteins, but not very many of them. Some of these vesicles are going to be going to lysosomes. Some of these vesicles are going to be going back to the endoplasmic reticulum. Some of these vesicles are going to go back to the nucleus. Some of these vesicles are headed for the cell membrane, or the cytoplasmic membrane. They're all different locations, just like they're all different addresses, right? And different proteins need to go to different places to do different things. The ones that are headed to the cytoplasmic membrane, which is the ones they always show you in the textbook, especially in this chapter, they go to that membrane, and then they're going to fuse with it, and the contents of the vesicles are going to be dumped out. And then those proteins can go do lots of things. They can go to different cells, they can perform as hormones, they can cause other events to happen in a different part of the body. So it kind of depends on what the function of the protein is, and that's going to depend on what cell is making that protein, right? And we are certainly at this point just talking about generalizations, right? So if we were talking specifically about a liver cell, I would list some very specific functions of the Golgi that might be a little different, and locations where things are going to be sent, which will be different than if we were talking about a skin cell, right? And you also asked about the lysosome stuff, so I'm going to put that over here. But I can't spell it. So the lysosome, again, very complicated depending on what kind of cell you are in. Single membrane. By that we mean, remember, that means one phospholipid bilayer. Right? That's what single membrane means. A double membrane, like around the nucleus, that's 2 layers of phospholipid bilayer, right? The lysosome is filled with enzymes. This is a little ahead of us. Enzymes are proteins that have a specific function. People talk about the lysosome as a digestive compartment. That's because the enzymes in there, the proteins in there, can break large organic molecules into smaller organic molecules. I'm going to put monomers to help us remember things from chapter 5. Things that can break down lipids, things that can break down carbohydrates, things that can break down proteins. Right? Because of that, the pH in the lysosome tends to be acidic. Because acids can break hydrogen bonds and interfere with hydrogen bonds better than bases can. And so that tends to aid that digestion or breaking apart. So the main function of the lysosome is to clean up, so it can digest things the cell no longer has use for, break things down back to the monomers that then can be reused to make different proteins, carbohydrates, lipids, as well as to break down things the cell may bring in specifically for digestion. So lysosomes we often see in conjunction with phagocytosis, right? Which is when a cell basically engulfs we refer to it as a particle. So either a large, let's say a large organic polymer, or another cell, meaning usually an invading type of cell, like a bacteria, a prokaryote, or a protist even, right? And then that vesicle that engulfed the particle, whichever it is, right? A food particle, that would be a large organic polymer, or an invading cell, a bacteria or a fungus, right? Or a protist. That vesicle is then going to always fuses with a lysosome. Right? And so when you look at chapter 7, you'll see in endocytosis, and particularly phagocytosis, that's when you're going to have these vesicles that are going to fuse with the lysosomes, and the lysosomes then will release those enzymes inside the larger vesicle that will digest whatever it is. And then the cell recycles everything. It's going to use what's left over to make new items or use them for building blocks, save them for later. Better or worse, Steph?
Much better, thank you. And I have another question too.
Okay, go ahead.
I'm going out of order. I'm sorry to the classmates.
Quite all right.
So for microscopes, I wrote down from your lecture, bright field, phase, fluorescent, and both electrons. Will there be questions on the test regarding any of the other, like the cryo, or no?
Probably not. Maybe confocal is the only one that you didn't mention, and I may have left that out. It's hard for me to remember specifically. I usually spend a little time talking about confocal because it's how a lot of the pictures that you're going to see this semester, in the rest of the pictures, that particularly chapter 6 And then all the mitosis pictures, a lot of those are confocal microscopy, but I may have left it off on purpose because it was just too much. So I would just pay attention. If that's on the list, just know its features, which very simply, it uses it's really a variation of fluorescent microscopy. So you use fluorescent dyes to label the cells. And I'm going to put slash or parts of cells. And then rather than illuminating with UV light, you actually use laser light, so lasers, to activate those fluorochromes. So fluorescent colors. And then the image is actually, I'm going to put in quotes there, read or viewed with a computer screen. So that what happens is you can actually see different structures. So large structures like microtubules, microfilaments inside cells. And the other main advantage is that it's mostly how people literally watch with time-lapse images. It's how they watch cellular physiology. And I think this is all actually irrelevant. I think this is usually more a microbiology question. But for example, if you want to see how a muscle contracts, how the calcium is released from the sarcolemma, you can label the calcium with one color. You can label the sarcomere a different color. You can label the myosin heads a different color. You can label the ATP a different color. And you can actually watch or visualize the steps in a physiological process. The only reason that I'm mentioning it, it's kind of on my brain for Bio 1, is that when we get to the mitosis chapter, so cell division, so cell cycle, you're going to actually see a lot of confocal microscopy, particularly in animal cells. So all of those animal cell images, which I don't know the page offhand, all of those images are actually taken with a confocal microscope. So this is probably irrelevant. So sorry, I just kind of rambled on there. I don't think there are any questions about it on the test.
Thank you.
You're welcome. It's hard to resist, especially like when I'm in a classroom in front of a board. You know, it's like, oh, so many things I want to tell you about biology that I can't. It'll last. All right. Does anyone else have any questions, comments, worries? Peroxisomes. So those are, peroxisomes are a specialized, they're actually kind of like a specialized form of a lysosome. I don't know what happened to my thing here. Where are my things? My controls. I don't know why I did that. So peroxisomes, someone asked in the chat, Phil asked in the chat, the peroxisomes are filled with hydrogen peroxide, hence their names. And they are, the picture in your book, I believe, shows them digesting a used mitochondria, an old mitochondria. This is not something that you have to spend a lot of time on. You should know the basics. Single membrane, so one phospholipid bilayer, hydrogen peroxide, and used to digest old organelles and large particles in conjunction with lysosomes. That's it. Don't obsess over too many of the details. Use, right? So use the note outline and add the videos from the details from the lecture videos, and don't spend 5 hours studying how a microtubule actually causes a flagella to move. I'm not going to ask you that. If it's not in the video, and it's not on the outline, I'm not asking it. It's a waste of time to spend your time studying that. Yeah, what happened to, there they are. Lost my controls. There we are. Better. Okay, anything else? Yes, no, maybe so. Just as an FYI to everyone, I'm going to try to announce this now, and I think I put that little piece of paper. So a lot of people are having trouble keeping it straight, like not particularly those of you that are actually coming to the Zoom meetings, but I understand, I get it. My office hours are at 11, and I'm also teaching a microbiology class at 11 o'clock. And so some of my students are having a hard time. They're trying to join like my microbiology class because they think it's an office hour on Tuesday, but it's not. It's actually my microbiology class. So my plan is I'm going to be deleting the office hour meetings and putting them in my personal Zoom room. And then I'm just going to post my Zoom room ID and the passcode for that. So office hours will just switch to the Zoom room. They won't be scheduled as meeting times. So those are going to disappear when you open up your Zoom page and you're used to seeing office hours tomorrow, today at 11 o'clock. I'm not going to do it until after this exam because I'm giving you guys an exam on Wednesday and I'm giving a microbiology exam all day tomorrow. And I don't want to add to the confusion, but I want you to be prepared because like I said, there'll be someone trying to join my 11 o'clock meeting tomorrow thinking it's an office hour and it's actually like the microbiology lecture and they're confused and I'm confused. I'm like, why, wait. And they're like, can you talk about cells? And I'm just like, what? No, that's not on this test. So I'm in the process of doing that. And I wanted to warn you ahead of time. The change will probably happen late this week, Thursday, Friday, maybe over the weekend. The other thing is sort of as a general announcement, you guys that are here, obviously you're doing great. I can see you, I can see your names. Please try to avoid joining the meeting with someone else's phone that has an identifier that I can't tell who it is, right? So it's clear, I've tried to make it as easy as possible to join, but for security reasons, I really do like to see your names because then I know that you're actually people that are in my class, right? I have people trying to join my meeting, like with someone's iPhone, for example, or their iPad that's named, like Prince Bob's super duper iPad. And I'm just like, okay, I don't know who you are, right? So I'm not going to let you in. And then I get an email. I tried to join the meeting at 10:30 and I couldn't get in. I'm like, yeah, because I had no idea who you were, right? So Zoom has made a lot of improvements to security. That's part of the reason why I had the waiting room enabled to begin with. And I was like, okay, I can take that off because only people are joining who I know. And then all of a sudden, in one of my other classes, all these people started joining, not all of these, it was 2 people with iPads or iPhones that had completely different names than the person. So that's me preaching to the choir, so to speak. This 4 or 5 of you that are here, obviously you're not the problem there, but I'm saying that so that it'll be in the recording so that other people, without me calling them out specifically, will know that they need to either rename those devices or use their own device. Okay. Other content questions, 6 and 7? Anything from 7? Osmosis, passive transport, diffusion, facilitated diffusion. Yes, no? All right, then. I'm going to call it a meeting. Good luck tomorrow. And just keep an eye out. I did send an announcement, I think, that the lab, because some people still are just getting their lab kits this week, the lab that was originally due last Friday, I moved it to Wednesday. So if you didn't get it finished, you can still do it and get it turned in time. Please, please, please remember, I need a picture of you, your face with your results, right? And filled out the lab report. So I had best intentions to make those lab reports a little more involved, and I just gave up. So they're just posted and they're published, and you can just fill them out and turn them in. Yay. Other questions, comments, worries, concerns? Alrighty then.